I was recruited to Citran to work on motor neuron disease and ALS, frontotemporal dementia. And the idea there was to actually use all of the knowledge I had acquired in RNA biology and biochemistry. And I knew a lot about the RNA metabolism, how it works in a normal cell, in a physiological condition. So was to see, if I could apply that knowledge to disease neuron in order to identify what events of altered RNA metabolism would be causing disease. So this project, as I was saying, has been sort of part of a very big gene therapy program that we have in the lab. Now, what we also found is that when we reduce the level of this transporter, we are also changing a few other RNAs in the process. So what we are now aiming to do in this grant is we want to understand in detail the molecular mechanism. So how does this work, and how does reducing the level of our transporter leads to reducing the TDP-43 pathology? So what gene therapy does is it's working on the expression of some RNA molecules that can be used. So they can be used through a virus, which is usually called an adeno-associated virus, which is non-pathogen for human. So we can manipulate this virus that will then lead to the reduction of our protein of interest. But what we can also use is directly on oligonucleotide. So an RNA oligonucleotide that can directly be adding to the culture, let's say, of our cells, or hopefully later on directly injected into a patient. And this is, for example, an approach that was taken for SOD1 with a drug which is called tofersen, and has yielded to really exciting result in clinical trials. What this will allow us to do, this grant, is if we can show that reducing the level of the SRSF1 proteins promotes indeed the motor neuron survival in 20 different ALS cases, in 20 different lines derived from patient, this will actually enable then very quickly, hopefully, a second clinical trial, because all of the tools and all of the safeties would already have been developed by Crucible. So we could hopefully, get very quickly into a second trial. And this would obviously be of interest to the company as well. So we would directly have a partner to then move from this discovery research phase to the clinical trial. So what I hope is that we will achieve all our aims, which are indeed to show that reducing this transporter confers neuroprotection to the 20 different lines of patients, but also that we can understand the mechanism by which it's doing this. So we have one of the aims in this project is to sequence, so to identify all of the RNA we have in a cell. So that's about 200,000 RNA are made in a cell. So we will look at all of them individually and can really understand the mechanism at this detailed level. And obviously, my overall aim is that this leads to a clinical trial that obviously would be, all my focus basically for the next few years through C9, but then hopefully through sporadic cases as well, and all TDP-43 proteinopathy. By the end of this project, we will have the proof of concept that reducing this protein is a novel gene therapy approach for patients with TDP-43 proteinopathy. This research should be going quickly to the clinic because there is no true model of sporadic disease. So we wouldn't need to do any test in animals. But also, as I've just said, we already have Crucible Therapeutics working on developing the clinical drugs, either viruses or interfering RNA that would be ready to be used in the clinic if those are safe in the current study we are doing with Crucible Therapeutics. Potentially within 5 years maybe, or this research could be in the clinic.